We've talked about enzymes before. These are in the class of proteins, and we looked previously at kind of a simple look of enzymes where a substrate enters the active site, the enzyme facilitates a reaction in its active site, and products are formed. And this is how enzymes function, right? Enzymes make chemical reactions that would take forever possible on biological timescales, right? It's possible this substrate could break apart on its own, but with the enzyme, it's able to churn through this reaction and have it occur when we want it to. So enzymes as reaction facilitators is review, and this is the basis to what we're going to be looking at here. Because we know this is how enzymes work, but we don't want enzymes working all the time, right? If an enzyme is making a reaction happen, do we want every reaction that an enzyme corresponds to be going at 100% all the time? Not really, right? We want enzymes to do a function that our bodies need when we want that to occur. So how is the body able to control enzymatic action? How does the body turn on and turn off enzymes? And so this is discussing enzymatic control, right? How do we see how bodies control the function of enzymes? Enzymes are very specific. Each enzyme goes with a certain reaction only, and we've looked at this naming already. Cofactors are metal ions, organic molecules. These are things that are required for an enzyme catalyzed reaction to occur. Basically, this is one way to control an enzyme, is it only works in the presence of some second molecule. Or a regulator is something that can control enzyme activity by turning up the activity or turning it down. We're going to see how this works as well. So here's an example of a cofactor or a coenzyme where we have the active site of the enzyme and it only fits the substrate once the cofactor is there, right? In this example, the cofactor is going into the active site, and that basically makes the active site have a shape that can accommodate the substrate that it's targeting. This is one method of control, right? Because if the cofactor is not there, the enzyme's off. It cannot catch the substrate. If the cofactor is there, then the enzyme can catch the substrate because the active site fits the pocket. Another way is through inhibition. So this is kind of like the opposite, where we have an example here of a competitive inhibitor, where the substrate would be fine to fit into the active site of the enzyme, except this competitive inhibitor comes along and blocks the substrate from entering the enzyme active site. This prevents enzymatic action because this competitive inhibitor, which is not part of the reaction the enzyme catalyzes, is blocking the substrate from going in there. This could also be through non-competition, and so now we're going to look at this allosteric site, this site that changes the shape of the enzyme. That's what we should think of when we see allosteric site. The non-competitive inhibitor, when bound to that allosteric site, changes the shape of the active site and still has the same effect. The substrate can't enter. But remember that these enzymes are these large globular proteins, and if we shove a big clump of non-competitive inhibitor onto the back of the protein, it could change the shape of that active site so that it no longer is able to attach to a substrate, right? This is two ways that we can inhibit enzymatic action. We could also have allosteric control through activating, right? Say the enzyme has an active site that doesn't work with the substrate of interest, but only works when the activator attaches to that allosteric site, right? In the right example, when the activator attaches to the back of the enzyme, again, it shoves the enzyme into some new conformation, but only when that activator is there does it activate the substrate. We call this allosteric activation. So again, we see that the presence of a secondary molecule can either inhibit enzymatic action or activate enzymatic action. And this altering of the active site may not completely turn off action. It could just promote or reduce action, right? It has degrees of control that these activators or inhibitors can perform. A good example of, enzymatic change is penicillin. So penicillin is an antibiotic, we know it pretty well, kills bacteria, but it doesn't really harm humans, right? And how does it do this? Well, it kills bacteria because it irreversibly binds to the enzyme that creates the bacterial cell wall. And therefore the bacteria can't reproduce. It can't create new bacterial cell walls, and therefore, our immune system takes care of the bacteria as there's no reproduction happening. No effect on human cells because human cells don't have a bacterial cell wall, so it doesn't change that mechanism. And so this is why penicillin has kind of a target on all bacteria, right? It's an antibiotic. One of the challenges with antibiotic, though, is that as we use more and more of this, bacteria is smart and it adapts. So it could figure out how to use enzymes that don't respond to penicillin in order to make bacterial cell wall and persist. That's a bit of a different issue, but the point here is that it is through enzymatic control that penicillin is able to target bacteria specifically while not targeting human cells. Finally, we can have really complicated feedback mechanisms for enzymatic action. So this is an example of such a thing, right, where we have a substrate that goes with an enzyme that makes some new things and makes substrate B, and so we have a few enzymes that go through to create an end product. And it's conceivable that this end product could be an inhibitor of the original substrate enzyme 1 reaction in the first place. So it could be, and it is very often in biochemistry, that we only want the enzyme to work until we have enough of the final product. Once that final product is made, it can turn off the enzyme. If our body is using that final product and eventually there's not much of it around, then it'll fall off of the enzyme because it's being consumed and the enzyme will be active again. So, this is really a testament to the structural diversity of proteins, that they are able to have this enzymatic control where secondary molecules can turn on, turn off, promote, reduce the function of the enzyme, and that the enzymes are so inherently important to biochemical reactions that we pretty much have these facilitations that are run pretty much just by proteins.